The next thing I want to talk to you guys about is your dependent and independent variables along with constants and controls. So let's get away from the actual science, you know, planets, moons, all that. And let's talk about experiments and things you need to know about dependent, independent, constant, and control. So I'm going to do it more of a scenario way. I'm going to draw it out into an experiment so you can see how it works in real life. Okay. So let's say that I am testing fertilizer. All right. And I want to see if fertilizer A is better than fertilizer B. Okay. So let's say fertilizer A is the designer brand and fertilizer B is the Home Depot brand of fertilizer. And I want to see if the Home Depot brand is just as good as the designer expensive brand. Okay. So what I'm going to do is I'm going to take 2 pots. And these 2 pots, even though they don't look like it because I'm a terrible drawer, but they are exactly the same. Exactly the same. They are made of the same stuff. They are both terracotta. And let me move this one over here just for a second. Make them a little bit closer. They are both terracotta. They're both made out of the same substance. They're both the same size, exactly the same. Okay. Then I add dirt, and they both get the same amount of soil, exactly the same type of soil, everything. Okay. Then I add my seeds. Same again, same type of seeds, same amount of seeds. Everything is the same. Okay. Nothing is different. And then I make sure, oops, that is not the color of the sun. Then I make sure that they both get the same amount of sun or light. If I'm growing them indoors, they both get exactly the same amount of sun, totally the same. I don't put one in the shade and one in the dark. They're both getting exactly the same amount of sun for exactly the same amount of hours in a day. Okay. The other thing I do is I water them. They both get the exact same amount of water every single day. The only difference is for this pot here, I sprinkle a little of fertilizer A, and for this pot here, I sprinkle a little bit of fertilizer B. The amount is the same. Let's just say I do a tablespoon for this pot here of A and a tablespoon of fertilizer for this pot B. I mix it exactly the same. You know, I do everything the same. The only thing that's different is one is getting A and one is getting B. Okay, so let's analyze that really quickly. What are my constants? My constants are the actual containers, both terracotta, both are the same size, samesies. The amount of dirt is a constant. The amount of seeds is a constant. The amount of water is a constant. The amount of sunlight is a constant. Why is that important? Because I need to figure out if the thing making the change is the fertilizer. If I put one of these in the dark or in the light, or I water one more than the other, how do I know that it's not the water that's making it grow and not the fertilizer? So if you're testing fertilizer, you have to make everything else identical. All right. So your constants here are all those things we just talked about. All right. Now let's talk about the independent variable. The independent variable is the thing the scientist manipulates. In this case, the independent variable is the fertilizer. That's the thing the scientist does to the experiment. So for example, if I was testing drugs and I had 40 people in a room and I gave them a drug, the drug is the independent variable. Everything's the same in the room. The temperature is the same. We gave them the same type of lunch. We fed them the same food. We gave them the same amount of water. The only thing that was different was I gave them a drug. That's the independent variable. In this case, the independent variable is the fertilizer. Those are the independent variables. Okay. Now, as I wait, my plants start to grow. Takes a little time, but my plants start to grow. And it looks like fertilizer B is really growing. The generic, we'll make sure we remember that this is the generic brand here. So it looks like when I measure, and let's do a little measurement here, take my ruler and I see that my second plant that's using the generic fertilizer is growing much more quickly at a higher rate. So it looks to me like fertilizer B is the winner here. What is the dependent variable then? The dependent variable is the difference in growth or the height of the plant, because the dependent variable depends on the independent variable, the fertilizer. So this is very important you remember this when you're answering questions on the test. They will ask you a dependent variable question or an independent variable question. All right, so that's the dependent variable. Now, they may also ask you something like this. What would make this study stronger, better? Well, it would make it better if you added a control. A control is the same thing, Everything is the same. I am watering it the same as I did the other ones. It's getting the same sunlight, even though it looks like it's not because it's over to the side, but same everything. Everything's the same. The only difference is no fertilizer, none. No fertilizer. Because then what happens if this plant goes bigger than all of them? Now I know it's maybe not the fertilizer and I have to do another experiment. Or if that plant only grows that much, then I know that fertilizer is working and that B is better than A. So a control is what you measure it against. And you make an experiment stronger by adding a control. By just having A and B here, these two pots, it's okay. I can see if B is better fertilizer than A, but I need that control to measure it against. How much better is it than no fertilizer? And then how much better is it than fertilizer A? Now, another thing to help this particular experiment is its sample size. I'm only using 2 plants here, or 4 seeds in each thing. We should have hundreds of plants in order to make this stronger of an example. Sample size, the bigger the better. Bigger the better sample size. And you would also want to do it again and again and again and again and again, right? Because part of the scientific method is doing experiments over and over and over again to see reliability. And if you have reliability, it means you're getting the same results again and again. So if I do this experiment 10, 20, 30 times, and again, fertilizer B outperforms every single time, well, then I can say fertilizer B is a better product. But if I only did it once, I don't know if that's really the case. So make sure you understand that when it comes to the scientific method. And again, think about it as a teacher. You want to teach students how to do this in science class. Otherwise, they're just kind of doing labs and things like that. Oh, it's fun. But if you're really teaching them to be scientists, they have to think like a scientist. So the way you may see it, were you guys at my Facebook Live last night? Because if you were, I'll do a different question, but this is a good question. Did anybody see the Facebook Live yesterday at 3 o'clock? Let me know in the comments if you have really quickly. Good. All right, Darren has not. So I'm going to do this question because it's a good one. I like this question because you'll see it like this on the exam. All right. So you have this big old narrative here. And then you have your answer choices here. The first thing I want you to do when you have something like this on any exam, do not start up here. No, we don't start at the top of a question. You always want to look at your answer choices first because it kind of helps you understand what's going on. And then I always like to look at the question stem at the bottom of a big scenario. Because if you start up here at the top, you have something in your mind and then it's in there, and then you're listening or looking at answer questions, and now you got a bunch of information in your mind. It's better to work backwards and think about the task first and then go in. So that's my 2 cents. I know these tests very, very well, and reading the answer choices first always helps. Now, for a science exam, you may not always be able to do this because, like you saw the questions in the book, it may just be a quick definition, like what's the summer solstice or that kind of thing. But when you have a question like this, it works really well. So I've got here, each Daphnia should have its own tank. The length of the experiment is too long. There are too many independent variables, and the sample size is too large. All right, just by looking at the answer choices, I know right away that this is a nature of science question, and this is about experiments. So I want to think that in my mind. Can I eliminate anything right off the bat? Well, D can be crossed off immediately because sample sizes can never be too big. The bigger the sample size, the better. Which would you rather? I tested this drug on 2 people and it worked. Here, take it. Or if I said, I tested this drug on 1,000,000 people and it worked for 85% of them. Which one's better for you? The second scenario. So sample sizes can't get really big enough. We want as big, the bigger the better, all right? The other thing I can eliminate here is B. The experiment is too long. Some experiments take years and years and years and years and years to complete. So that's not something you want to take a look at. And then if we look at C, notice that I have these words that are associated with experiments, nature of science, independent variables. So I want to keep an eye out on C, but I don't want to eliminate A right away. All right, so this is this kind of test strategy I teach people. Teach your students this too, and your kids if they're taking exams. So now let's take a look at the question stem. Which of the following is a major flaw in this experiment? All right, we're looking for the problem here. So let's go ahead and read. A student performed an experiment to determine the effect of temperature and light on the feeding behavior, of Daphne magna. So it's a plant. The student placed 25 Daphnia in each of 2 identical glass bowls. So we have our constant there, so that's good. And then placed one bowl in a sunny, warm window and the other in a cooler, dark closet. The student measured the amount of food consumed in 1 week. Which is the major flaw? Well, let's say I don't know. It's too much for me. I don't know. Go with the answer choice that has the words associated with nature of science, because you know this is a nature of science question. I'm going to go with C because it has that independent variable, and that goes with nature of science. But C is also the answer because you're determining if temperature and light affect feeding behavior. You can't do 2 because you're not going to know, is it light or is it temperature? You need to measure things one at a time. You need to apply things one at a time. So in this case, there are too many independent variables. There are too many things that you're doing to this plant, and you don't know which one is having the effect on the feeding behavior. So that's why C is the correct answer there. So that's a nature of science type question. You will be asked things like that. You may even see a question just like this, only adapted differently. It might be not a plant, it might be a mouse or something like that. Just know that you want to have only one independent variable, and you want to make sure you have your constants and your controls to make it a strong experiment. Now, let's say she did all the things right. The student did all the things right. And it said, which of the following would make this experiment stronger? Pick all of the following. Well, you could pick add a control. You could pick do it again and again, repeat the experiment. You could pick make a bigger sample size. All 3 of those things would make this experiment stronger. Also would make this experiment stronger to take off one of the independent variables and measure them. Do one experiment for temperature. Do one experiment for light.